All right, you guys, I'm glad you came back for the last few slides. We're talking about receptive fields within the visual cortex or the striate cortex. And we said earlier in lecture that we can think of them as receptive fields for now while we're learning this, but because they're more complex than receptive fields, we're going to give them a new name, which is orientation selectivity or orientation specificity. Okay, so there's lots on this slide. Let's start with what we know. This should look familiar over here. You've got center surround receptive fields, which you know are associated with retinal ganglion cells, right? You would correctly refer to the receptive field of a retinal ganglion cell, either an M cell or a P cell, depending on what type of information this receptive field is gathering. And we said earlier in lecture that there's pretty much a 1-to-1 ratio from a retinal ganglion cell, which is the primary neuron in this pathway, and neurons within the lateral geniculate nucleus, which are the secondary neurons in this pathway. One retinal ganglion cell projects to one neuron within the lateral geniculate nucleus, one thalamic neuron, and the thalamic neurons are the secondary neurons in this pathway. And then in the first part of this lecture, we said that the receptive fields of the LGN thalamic neurons mirror the receptive fields of the retinal ganglion cells that project to them. So that's depicted a little differently here because we're now talking about the secondary neuron, but there's still a center surround receptive field within the thalamic neurons. Okay? Hopefully you're good with that. If we were in lecture, I'd ask you if that made sense. I hope that makes sense. One retinal ganglion cell projects to one thalamic neuron within the lateral geniculate nucleus. The receptive field of the neurons within the lateral geniculate nucleus therefore mirror or are similar to the receptive fields of the retinal ganglion cells that project to them. These are the secondary neurons in this pathway, and they project to the striate cortex, visual cortex, V1. Now, within the layer 4 of V1, we have two different types of cells, simple cells and complex cells. Let's start with the simple ones. So this is an example of a simple cell, and it's receiving input from multiple LGN neurons. So the receptive field of this simple cell is going to be the sum of the receptive fields of the neurons that project to it. So let's look down here at the bottom left. So if these are the receptive fields of the retinal ganglion cells, which would be similar to the receptive fields of the lateral geniculate nucleus neurons, imagine what the receptive field would look like if we squished these closer together. Move them closer and closer and closer together. Squish them. And if you squish them, hopefully you can imagine how you would get a receptive field that looks something like this. Okay? So then, if we go back over here and read this, a simple cell responds best to a bar of light in a particular orientation. In this example, it's a horizontal bar of light because of the orientations of these specific receptive fields. Could it be vertical? Could it be horizontal? Could it be vertical? Yes. Could it be diagonal? Yes. It'll depend on the receptive fields of the retinal ganglion cells and LGN neurons that are projecting onto the simple cell. But these are called simple cells because it's fairly simple. If you were able to follow me through that, it's pretty simple to predict the receptive fields, which we're now going to call orientation selectivities, of simple cells within V1. Okay. Similar idea shown over here, but we've got, instead of an on-center off-surround, we've got an off-center on-surround. Same idea. And then things get more complex when simple cells project and converge onto complex cells, which are other cortical neurons within V1, and we're not even going to try and predict their orientation selectivity. We'll just say that it's going to be more complicated. Now we're going to take a bunch of simple cells, which may or may not have similar orientations, And then the receptive fields, or the orientation selectivities of the complex cells, will be a sum of the orientation selectivities of the V1 cells, or the simple cells that project to them. Okay, so I'd just like you to know that complex cells are more complex. So here's an example of what that might look like. Right? Here's the receptive fields of the simple cells, which are the sum of the receptive fields of the LGN cells that project onto them. So this might be reflective of each of these simple cells. And then if we were to predict the orientation selectivity of the complex cells, we would have to figure out how these might add up. And we're not going to do that other than to say that it's more complex. You need to know that complex cells, which are found in a number of regions in the visual cortex, B1, B2, and B3, they receive input from multiple simple cells that, in this example, have similar receptive fields, but that's not necessarily true. And then hopefully you can imagine how complex cells then may exhibit directional specificity in terms of light moving. So rather than just a horizontal bar of light, but a horizontal bar of light moving to the left, compared with a horizontal bar of light moving to the right. So a band of light or an object moving in one direction but not the other is often a property of the complex cells. Okay, but if that much makes sense, then you're good. All right, and then we talked, I described orientation columns as in here you can actually see them as columns, and each of these columns would contain cortical neurons in V1 or another region of the visual cortex that respond to input that is similar, i.e., a bar of light in the vertical direction, a bar of light in the horizontal direction. You can see these little lines here denoting the sensitivity of one column versus its neighboring column. And then the I and C is ipsilateral and contralateral. So in the first part of the lecture, we said that orientation columns contain neurons that are similarly sensitive to input that's coming from a similar place. So in the visual system, that means the right eye versus the left eye, and that means perhaps a bar of light in one direction versus another. Okay, that's really all orientation columns are for now, but you'll learn more about them in 444 if you go on and take that course. And then in the context of orientation columns, there are some orientation columns that are also concerned with color. And similarly to how we added up the receptive fields of the retinal ganglion cells and LGN cells that they project to get the more complex orientation selectivities associated with simple cells and complex cells, the same is true here. We can make it even more complicated if we consider cone opponency, which we talked about last time, and color vision. So one of these columns may be dedicated to, remember we said that red and green are cone opponent, right? They're color opposites. So over here in the corner we have, say, a red on center, green off surround. And what might the orientation selectivities of the cortical cells that are part of this pathway, what might they look like? And this diagram over here is showing what the firing rate in the cortical neurons that are part of these or one or more of these orientation columns that are specifically concerned with color, what might the firing rate look like in when presented with different types of stimulus. So here, a single red band of light, you get a little bit of firing. A green band of light, you don't get any firing. Don't worry about what's happening over here. It's beyond our scope. If you get 2 bars of red light, you don't get any firing. 2 bars of green light, you get a little bit of firing. And the best possible combination here is to have 2 bands of green light sandwiching a red band of light. And down here, you can see the opposite combination where you have 2 bands of red light sandwiching a green band of light, and that gets you no firing. So the maximum stimulus here is a red band sandwiched by 2 green bands. And hopefully if you apply what we were talking about on the previous slide, where we were summing up the receptive fields of the retinal ganglion cells and squishing them together, where you get that hot dog kind of shape and preferentially sensitive to a band of light in the middle, apply that same principle here and imagine squishing 3 of these guys together and getting something that looks like this, where this would be this would result in the maximum firing rate of those cone opponent or double opponent, as they're referred to, double opponent cortical cells. Okay, so double opponent cortical cells are responsible for color, and they will have orientation specificities that are similar to, or orientation selectivities that are summed, similar to what we discussed on the previous slide. And so the key point is they respond differently to different color combinations involving cone opponent colors, right? Red and green versus blue and yellow. Okay, and then other features or characteristics associated with visual processing that occur in V1, things like what's happening where, right? The ganglion cells will extract the edges, right? We said borders and edges where dark meets light. Higher cortical cells, like the simple cells, will extract the lines, the corners, and any motion if this was a moving image. And then when you put all of that information together, ultimately what you perceive is this image here. And again, the 2 supplementary files are really good for alternate explanations of this material. Another example of feature extraction, if we were in lecture, I'd put this up and ask you if you saw anything, and some of you might see something in here. You may see giraffes, and if you can't see the giraffes, here they are. And of course there are no giraffes in this image. There are just borders and edges where dark meets light. But feature extraction is pulling pictures out of an image. And once, of course, you've seen the giraffes, you can't unsee the giraffes if you're presented again with the black and white image. Feature extraction as it relates to color. I can't see anything here. Even if I'm looking closely, I see a bunch of shapes, but nothing in particular jumps out unless we add in some color. And this is perhaps not done best in red and green, because anyone who has deuteranopia or protanopia won't be able to see this, but those of you who can see the difference between red and green should hopefully see color spelled out in American here. It's missing the U, but C-O-L-O-R. So color vision likely evolved. There's a number of different theories as to why color vision evolved, but most likely it was so that our ancestors could determine which fruits were ripe, red fruit against a green background, and so on in terms of foliage, in order to determine which fruits were the most nutritious. So that's one theory. There are others. All right, I will leave you with some review questions and encourage you, as always, to stay in and stay safe. I hope you're listening to Dr. Bonnie Henry's updates each day. I think Saturday, tomorrow, will be at noon, but the news was good today. It was at least less grim, and it seems like the measures that we're taking, particularly in BC, are starting, it seems, to have an effect. So stay safe and stay well, and keep doing what we're doing, staying in, and I'll talk to you guys on Monday.